So firstly, my name's Charlotte McNamara and I'm the head of the health department at the Kennel Club. We are really excited and pleased, if not a little bit nervous, to be offering our first health webinar online this evening. I'm really delighted that Dr. Joanna Ilska, who now works for the Kennel Club, is providing our webinar this evening on the science of DNA testing. Joanna joined us in January 2021, not 2001, 2021. And we're really thrilled at the expertise and the ideas and the energy that Joanna has brought to the health team and the health department at the Kennel Club. We'd ask that you enjoy the webinar as you go along. If you do have questions, then please do add them into the chat or the Q&A section. We will try to answer as many questions as we possibly can. We have received some questions in advance, which we will try to cover. If there's anything that we're not able to cover or there's anything that's more specific, then we will come back to you and try to answer those individually after the webinar. So I'm going to hand over now to Joanna, and I really hope that everybody enjoys this presentation and webinar.
Thank you very much, Charlotte, for the introduction. I hope that everyone can hear me and you can see my presentation. We have quite a lot of material to cover today, so I'm going to just jump straight in. We are going to start right at the beginning with a short refresher on DNA and what mechanisms cause genetic variability. We are then going to discuss how this variation in genetic code is inherited across generations and how the DNA variability affects the health of our dogs. We will discuss why some mutations are unique to some breeds, while other mutations appear to be widespread across different breeds. Then, of course, we will move to breeding against monogenic disease. We'll start with the practices of the past, and we'll move to the gold standard of the DNA testing that we are promoting today. We will discuss how DNA testing works and how it can be used for dog owners and for dog breeders. We will also talk about how DNA testing results contribute to research on dog health. In the final part of this webinar, we're going to switch gears a little bit, and we're going to talk about the benefits of DNA profiling and how it can be used in parentage verification. So let us start right at the beginning with some basic definitions relating to genetic code. As most of you will know, our genetic code is contained in DNA, deoxyribose nucleic acid. DNA can be found in almost every cell of an organism, primarily in the cell nucleus. Within the nucleus, strands of DNA are tightly coiled into structures which we refer to as the chromosomes. DNA contains the primary unit of heritable information, the genes. The bits of DNA which are coding for specific genes are separated by what we call intergenic regions. And some of those intergenic regions have very important regulatory functions, while others appear to have no function, or at least this is as far as we know for now. Genes code for proteins, and proteins make us who we are. There are huge numbers of different types of proteins produced by our bodies, starting from building blocks through transport proteins, enzyme, antibodies, and so on. Some proteins are crucially important for the functioning of the organism. So, for example, hemoglobin is a protein which is responsible for transport of the oxygen in our blood. So obviously it's essential for life. While other proteins, for example, pigment proteins like eumelanin, have slightly less important functions. In most cases, they just affect the appearance of the animal. A DNA strand is formed from nucleobases. There are 4 bases: adenine, cytosine, guanine, and thymine. While looking at the string of ACGTs may seem not very informative to us, the order in which those nucleobases are actually determines the genetic code, and so it is crucially important. A sequence of 3 nucleotides forms the basic unit of the genetic code, and we refer to that as codons. A codon codes for production of a specific amino acid, and several amino acids form a protein. So each DNA molecule of a dog contains 2.5 to 3 billion bases, and as we've said, they are in a very specific order. We've also said that nearly all cells in a body contain DNA. So that means that every time a new cell is created, be it anew when the organism is developing or as a maintenance when new cells replace older ones throughout our life, the DNA in that original cell needs to be replicated. This means that the 3 billion bases of DNA have to be replicated millions of billions of times throughout the lifetime of an organism. And this process of DNA replication is, of course, very efficient. It must be, given the importance of the DNA molecule. However, when something is replicated millions and billions of times, there will be small errors. And these small alterations in DNA codes as a result of replication errors are what we call mutations. There are a few main categories of mutations. The most common ones are when a specific base is replaced by a different one. So here in this example, we see that a guanine was replaced with a cytosine. We refer to these mutations as substitutions, and in the genetic jargon, they're quite often just termed as SNPs, so single nucleotide polymorphisms. Another type of mutation includes an addition of a base. This is called an insertion. In this example, we see that a cytosine has been added in between a trio of guanines and an adenine. Equally, a mutation can result in a loss of one or more bases. This type of mutation is called a deletion. In this example, we see that we have lost a couple out of the trio of guanines. So insertions and deletions can involve one or more bases, and further, these types of mutations tend to have a larger impact on the protein because we are changing the reading frame, so the codon is changing. It affects not only the codon where the mutation took place, but also the following codons. So, of course, what we are mostly concerned with when we consider mutations is what is the effect of the mutation on the protein. And this can be predicted to a certain degree. We know that genetic code is unambiguous, so that means that each codon only codes for one amino acid, but it is also redundant. The same amino acid is coded for by multiple codons, and this is what we can see in this table where we have all possible codons, combinations of ACGTs, and the amino acids that they produce. So if the mutation changes a codon, but the resulting amino acid is the same, then we are predicting that there will be no effect on the protein. So for example, if the last base in a TTT codon is replaced with a C, the resulting amino acid is going to be exactly the same. It's going to be phenylalanine. We call this type of mutation mutation synonymous, silent, or neutral. However, other mutations will cause a change in the codon, which will result in a change in the amino acid produced. For example, here a mutation resulted in a cytosine being replaced by adenine, and as a result, the amino acid changed from phenylalanine to leucine. We refer to these types of mutations as non-synonymous. And those non-synonymous mutations are what we are primarily interested in. They can be advantageous, so the new mutated protein can be better than the original in some way. However, this scenario is relatively rare. In most cases, non-synonymous mutations result in some deleterious effect, from a slight lowering of the efficacy of the protein to catastrophic consequences where the protein is not produced at all. So when a mutation creates a new version of the genetic code, we refer to those versions as alleles. Sorry. So for example, GGG and GGC are two alleles of the same codon. Now, there are multiple ways in which alleles may be referred to. In the rest of the presentation, I'll use different colored squares to label different alleles. In scientific literature, the original allele is frequently referred to as the wild type, and it's often marked with a plus sign. And the mutant allele is identified with specific details of the mutation. In other publications, you may find alleles coded as capital and small letters, or even using N for the original allele and a specific letter for a given mutation. So, for example, N for normal and M for merle. So whatever the source, it is very important that when you're reading the genetic data, you know how the authors decided to code the alleles. So let's now talk about the inheritance of those mutations. New life begins when sperm fertilizes an egg. A zygote, which is formed in this way, contains the first copy of the complete genetic code an individual will have, and which is a unique combination of the genes inherited from the sire and the dam. The embryo then starts the process of cell division. At some stage, the cells start to develop into specific directions, into specialized cell types. We know now that all organisms will produce some completely new mutations as a result of DNA replication errors. For example, it is estimated that each human, every single one of us, possesses around 70 completely novel mutations in their genome, which they haven't inherited from their parents. But whether these mutations will be passed to their offspring depends on where these mutations appeared. If a new mutation happened very early on in the development, it will be present in all subsequently formed cells, including reproductive cells. We call these mutations germline mutations, and they will be passed to the offspring of an individual. If a mutation happened at a later stage and in non-reproductive cells, it is going to affect the individual to some degree, but it won't be passed to their offspring. We refer to these as somatic mutations, and they don't really have much importance in the context of breeding for health. So let's go back to germline mutations. Each individual has in its genome mutations that they have inherited from their ancestors, as well as the novel mutations that are unique to them. Dogs, just as humans, are diploid, which means that we have two copies of each gene, one inherited from the sire and one from the dam. The reproductive cells, the sperm and egg, are the only cells which possess one copy of each gene only. And this is so that when they merge into fertilization, the new embryo is again going to have two copies of a gene, one from the sire, one from the dam. A genotype describes those two copies that a dog has for a particular gene. Because we have two copies of a gene, we can have three possible genotypes. We can either have a homozygous genotype where both alleles are the same, so they can be either both yellow or they can be both green, or a genotype can be heterozygous where the alleles are different, one yellow, one green. When an animal is homozygous, all of his reproductive cells will carry the same allele. However, a heterozygous animal will produce reproductive cells which can carry either of the two alleles. As a rule, each sperm and each egg has a 50% chance of carrying a specific allele. As such, some of the offspring may inherit different alleles of a gene from the same parent. With conditions determined by mutations in a single gene, we can predict the genotypes in the offspring as long as we know the genotypes in the parents. So if both parents are homozygous for the same allele, we know with 100% certainty that their puppies can only ever inherit the same allele. If parents are homozygous, but for two different alleles, we know that each puppy will inherit one allele from each parent, and so they will all be heterozygous. If one parent is homozygous for the green allele and the other parent is heterozygous, each puppy will have 50% chance of being either homozygous for the green allele or 50% chance of being heterozygous. If both parents are heterozygous, each puppy will have 25% chance of being homozygous for the green allele, 25% chance of being homozygous for the yellow allele, and 50% chance of being heterozygous. So now that we are all on the same page with the terminology, let's move to the role of DNA in dog health. Dogs are like any other species, really, and their health is determined by a number of monogenic conditions, where the diseases are caused by mutations in a single gene, and a number of polygenic conditions determined by mutations in a large number of genes. On top of that, environment, of course, plays a role, particularly in polygenic disease. For the purposes of today's webinar, we are only going to concentrate on the monogenic diseases. Hopefully, in the future, there will be another webinar to cover the polygenic diseases. We know now about a whole number of monogenic diseases in dogs. In fact, dogs have the highest number of known monogenic conditions among animal species. The screenshot you are seeing here on this slide is from an online Mendelian inheritance in animals database, which is a great resource for anyone interested in genetics and particularly in monogenic conditions. You can see that there are currently over 300 monogenic traits for which a causative mutation is either known or is very, very likely. So that's a huge number. And there are several reasons for such a high number that have been described in dogs. Firstly, dogs are a very useful model species for human disease studies. We have a large number of breeds, with dogs being very uniform genetically within breeds and diverse across breeds. Secondly, we have comparatively large family sizes. And finally, dogs are our companions and share our environment like no other animal. So they are a very attractive species to study, and this results in a large number of scientific publications. However, there are also genuine reasons for the high number of mutations in dog breeds. The two major ones being the fact that dog breeds are close populations, which promotes accumulation of mutations. And we have evidence of the use of popular sires in pretty much all breeds. We'll talk about popular sires a bit later on. So where do mutations that we find in our breeds come from? First, there can be mutations which were present in the original founders of the breed. Those could be the mutations that were unique to those founders. They were novel, they arose in those founders, or they could be mutations which the founders inherited from their ancestors. Secondly, every dog used in breeding across the generations since the closure of the stud books could spread his or her own novel mutations. A mutation can, of course, be introduced into a breed through crossbreeding. It could be a result of legitimate outcrossing. So, for example, if a breed is in critically low numbers, it might be outcrossed to save it from extinction. But it could also be introduced by undetected outcrossing. This could be deliberate misregistration, but it could also be a genuine accident where a bitch we mated with a sire of the same breed has been later mated by a neighbor's dog of a different breed. If undetected, the genes of that neighbor's dog can then spread throughout the population. It is possible that some mutations which were present in the founders of the breed may have been lost over time. First, they could have been selected against, either through natural or artificial selection. But they could also be lost by chance. In genetics, we refer to this phenomenon as random drift. When a new mutation appears, typically it will be present only in one copy. So the original dog, which is the source of this mutation, is going to be heterozygous. That means that each of his sperm has 50% chance of inheriting the mutation. And the chance of passing the mutation is then halved in each generation. So his grandchild has 25% chance of inheriting that specific mutation. However, this is assuming that both the original source dog and his son had only produced a single offspring. The more offspring a dog produces, the higher the chances that at least one of his offspring will inherit that mutation. So this is where we are getting to the subject of popular sires. If all animals in our breeding population contribute equally to the next generation, then we are maximizing the chances of novel mutations being lost through random drift. Further, because we are maintaining all possible lines in the breed, it will take more generations before we reach a point where we are forced to breed related animals which carry the same mutations that they inherited from their common ancestor. On the other hand, if any single dog contributes to the next generation to a much larger degree than the rest of the population, so if we have popular sires, we are heading for trouble. As we've said, the larger the number of offspring any dog produces, the higher the chances that he will pass his novel mutations to at least some of them. And further, the more offspring he produces, the fewer the generations it will take before we will see this dog in pedigree of all dogs in our breed. And so we will be forced to breed together animals which will have him as a common ancestor. So this, of course, means that we are risking breeding two dogs which carry the same novel mutations that they have inherited from their popular sire ancestor. And unfortunately, dog breeding is heavily affected by the use of popular sires. We used to see winners of particular competitions, be it conformation shows, working events, become the sire of choice for large numbers of breeders. In more recent times, we see similar with dogs which have a particular trait. So, for example, it could be a new fashionable color. But worryingly, we also see that in some cases, dogs which appear to be clear from known monogenic conditions are being used disproportionately. The DNA all clear dogs. While these dogs are certainly very attractive in the context of those known conditions, we need to remember about those unknown mutations that are unique to these dogs that we are spreading throughout their unproportional use. And whatever the reason for the choice of a popular sire, it has been shown again and again that they are the primary reason for the reduction of genetic diversity and accumulation of the deleterious recessive mutations in dog breeds, even in open registry populations. Let's now go back to the mutations that are present in a breed. We know that different breeds will have different mutations based on the history of that particular breed. Sharing of mutations across breeds is thus dependent on the age of the mutation, so how long ago has the mutation first appeared, as well as the relationship between breeds, specifically whether there have been any crossbreeding events in those breeds' histories. So if a mutation appeared a long time ago, before breed formation, it can be expected to be present in a large number of breeds. A good example of such a mutation is hyperuricosuria. This particular mutation causes a disruption in the protein metabolism, which results in accumulation of uric acid in urine, which can then lead to formation of stones. It has been identified in a large number of breeds with no known recent crossbreeding events. And in fact, I recently tested remains of a dog found on a shipwreck from 1545. Showed that this mutation was already in existence 500 years ago. A mutation in GDNF gene appears to be far younger, as it has only been found in two related spaniel breeds, English Springer Spaniels and English Cocker Spaniels. This mutation results in a localized insensitive, oh goodness me, it's been a long day, sorry, insensitivity to pain, usually affecting extremities, so paws. And affected dogs will lick themselves excessively, which can lead to self-mutilation. We know that these breeds, those two breeds, separated relatively recently, and following the split, there was a considerable amount of crossbreeding that continued to take place. Based on the fact that this mutation has only been found in those two breeds to date, we can speculate that it was either a novel mutation which arose just before the breeds separated, or it appeared for the first time in one of the breeds following the separation, but was subsequently introduced to the other breeds through crossbreeding. And finally, we have a mutation causing leukoencephalomyelopathy, a neurologic condition in Leonberger, which appears to be unique to this breed only. So either it was a novel mutation that was present in a breed founder at the time when the stud books were closed, or perhaps it first appeared in a popular sire since the stud books were closed. So the advancements in the genotyping technology mean that now it is relatively easy and cheap to genotype large numbers of dogs across large numbers of breeds. And this has been done. There have been some large-scale genotyping studies which found that many mutations are present across many breeds, including those where the condition they are supposedly causing has not been widely reported before. Unfortunately, obtaining clinical information for those same dogs is considerably more difficult. So this begs a question: are we sure that a mutation that causes a disease in one breed is definitely going to cause the same disease in a different breed? And unfortunately, recent findings show that this is a dangerous assumption. When we test for a specific mutation, then we are only learning about that specific point in the genome which has been associated with the disease in a specific breed with the genetic background of that particular breed. But the different breeds have different genetic backgrounds. So let's say that a substitution of guanine with cytosine at a specific point in a specific gene has been associated with a disease in breed A. Subsequently, we find that exact same mutation in breed B. However, it turns out that breed B has a different mutation in a different gene that we are not testing for. And this second mutation may alter how the first mutation is going to behave. It could have a protective function, essentially making the original mutation a benign variation in breed B. Secondly, identification of specific mutations is a process which requires careful design in terms of the used samples. For example, in some cases, if we don't have enough dogs, we might pick up mutations which are actually benign, but by chance they seem to co-segregate with the condition. Secondly, if we are dealing with a breed with distinct populations in different countries, those distinct subpopulations may start behaving like different breeds. So they may start to develop different genetic backgrounds, which may affect the causality of the mutation. So as such, while the DNA testing technology is very exciting, we need to be careful with how we select specific tests that will be used to improve the health in our dogs. The health team at the Kennel Club has developed a procedure for evaluating tests before we start recording them in our database. First, we require that mutation tested for is published in peer-reviewed scientific literature. So this ensures that we have high standards of the research and the publications provide detailed evidence for the association between the mutation and the disease. Further, we require that before a test is recognized for a specific breed, the causality of the mutation on the disease is confirmed in that breed. We also examine the published evidence for the prevalence of the disease in the breed. While this isn't a limiting factor in terms of recognizing a DNA test, it gives us an idea on how high on the priority list for the breed this test should be placed in. So how do we add a new test? We are very much counting on the collaboration with the breed communities, as we know that in many cases you have the most in-depth knowledge of the issues in your breed. And of course, you will know about the ongoing research projects before they are published because you provide the data, you provide samples from your dogs. So we do reserve the right to recognize a new DNA test when our team, the health team at the Kennel Club, finds scientific evidence to support it. But in vast majority of the cases, we want this to be a joint decision with the breed community. So first, the breed health coordinators collate all the data that is available to them in terms of the dogs that have already been tested, maybe, or maybe any information about the research that is currently ongoing. And the BHC then fills out the request for DNA test recognition. This form is then reviewed by the health team, and we examine the scientific evidence in support of the test. If the test fulfills the criteria we have already discussed, we officially recognize the test, and then we start to record the results of this test in our database. All the results from that point onward can be accessed through our health test result finder. So as long as you know the registered name of a dog or his KC registration number, you can check all the health test results that we have in our database for this dog. In some cases, we find that the scientific evidence for the test is not complete. Quite often the mutation has been reported in the breed, but the causality of the mutation on the disease has not been verified. In such cases, we'll let the breed health coordinator know that we won't be recognizing the test just yet, but we are happy to reconsider when new evidence becomes available. And of course, in rare cases, we find that even though the test is offered commercially, Published scientific evidence indicates that there are some issues with it. Perhaps a new paper was published which showed that the original mutation was a spurious result. And in those cases, unfortunately, we will not recognize or record that test. So it may appear that we are quite strict with those tests. However, there are very good reasons for such control measures. When we are dealing with any closed population, and of course dog breeds are closed populations, it is crucial that genetic diversity is managed in a sensible way. Any sort of selection, be it for traits we find attractive in a dog or selection for health, reduces the overall genetic diversity. That's why we must be sure that the DNA tests that we use in selection are relevant in our breed. Otherwise, not only we're not going to improve the health of our dogs, but also we are going to unnecessarily reduce the genetic diversity with all the health risks and fitness risks associated with it. So now that we know how and when mutations first appeared in dog populations, let's talk about how breeders have tried to breed against monogenic diseases in the past. First, we need to differentiate between the genes the individual dog carries, or the genotype, and the phenotype, which is a measurable or observable characteristic of a dog. In the context of a disease, of course, the phenotype of a dog would be whether the dog suffers from the condition or not. In the past, breeders had to try to guess the genotype of the dog based on the phenotype of the dog himself or that of his relatives. The problem is that the phenotype and genotype are not necessarily equivalent. Their relationship will depend on the dominance hierarchy of the alleles at a gene and the effect of environment, which we'll ignore for today. Allele hierarchy considers how the alleles of a particular gene behave in reference to each other. Typically, especially in health conditions, one allele is dominant over the other. So if an animal has at least one copy of the dominant allele, the effect of the other recessive allele is completely masked. To see the effect of the recessive allele, an animal needs to be homozygous for that particular allele. For majority of the health conditions, the disease-causing allele is recessive. The other type of dominance hierarchy is termed as incomplete dominant, and those mutations, the mutations which are incomplete dominant will have some effect even if a dog has only one copy of the mutation, although the effect won't be as pronounced as when the animal is homozygous for the mutation. So let's consider a practical example. We have bought a healthy puppy after healthy parents, and in the future we would like to breed from our dog. Unfortunately, a couple years down the line, before we've had the chance to breed our dog, his litter mate was bred from and produced puppies affected with a recessive condition. So what can we do? There are several possible scenarios that people used to use in the past. First, we could wash out the line. We know that the litter mate who produced the affected puppies must be a carrier, and so must be the mate used in that breeding. But we also know that at least one of the parents of our dog and that litter mate must be a carrier too. Problem is that we don't know which one, or maybe both of them. This, of course, means that all the litter mates, including our own dog, are potential carriers. If we would like to wash out the line, we need to remove from breeding the whole family. But we know that this won't be enough, as the parents of our dog also inherited that mutation from somewhere. So technically, we would need to remove from breeding all dogs related to our litter, and of course, to the mate of our litter mate that produced the affected puppies. A second solution would be to perform a so-called test cross. So we could breed our dog, who we suspect that could be a carrier, to an already known carrier dog. If any affected puppies are produced, we will know with certainty that our dog must be a carrier as well. However, this type of test can only be reliably used when affected puppies are produced. So lack of affected puppies may mean that a dog is not carrier, but it could also mean that we just have been very, very lucky and we didn't produce enough puppies for our dog to pass the affected allele. So we would need to produce a large number of puppies before we are relatively confident that our dog isn't likely to be a carrier. And of course, Test crosses carry with them some ethical considerations, as we are knowingly risking producing affected puppies. The third solution is to only breed from dogs which haven't produced affected puppies. So we remove from breeding known carriers. However, this also isn't ideal, as we are potentially spreading the disease-causing allele through the use of carriers that haven't produced affected puppies just yet. So unfortunately, while some solutions have existed in the past, all of them carried risks. They took a considerable amount of time, and unless breeders were extremely lucky, those solutions seldom resulted in a permanent eradication of a mutation from a breed. This is because even with an extensive knowledge of a pedigree, breeders had to guess the genotype of a particular breeding candidate. Luckily, we are now in a much better position as DNA testing takes the guesswork out of the breeding against such conditions. As you will probably know, scientific world has been obsessed about genetics for the last 50 years, but the beginnings were fairly slow. The first bacterial gene was sequenced in the 70s, but it took 30 years for the technology to develop enough so that the first more or less complete draft of a dog's reference genome was published. It was a base-by-base sequence of the genome of a boxer bitch called Tasha. There were some gaps in that. We still have some gaps in the reference genome of dogs. However, the technology has come in leaps and bounds, and now it is relatively easy and cheap to generate the complete sequence of any individual dog we would like to examine. We just need a team of researchers, a couple of very expensive machines, and a few £1,000s to pay for that. But once the first reference genome was published, it became relatively easy to interrogate the genomes of healthy and sick dogs to identify the disease-causing mutations. So first, researchers select a group of dogs that are affected with a given condition, and then they add a number of healthy controls, preferably from the same breed. Then the researchers compare the genomes of sick and healthy dogs. We expect that the sick dogs will all share the same mutation, which should be absent in healthy dogs. And we can do that either by looking at the whole genome and then narrow down the mutations that we have discovered to non-synonymous mutations which fit with the clinical status in the animals. Or we can look directly at candidate genes. So those are the genes that we already know have a function in a specific disease process, either through previous from studies on other species, or maybe from studies in other breeds of dogs. This type of research typically leads to a scientific publication, and in recent years to development of a commercial test. So now let's go through the steps of DNA testing your own dog. First, you need to find out which tests are relevant to your breed. You can find this information in our Breeds A to Z guide on our website under Health section. You can either opt to order each test separately if there are multiple tests available for your breed, or you can decide to go for a breed bundle, which will cover all your required tests in one order. Then, of course, once you decide which tests you'd like to run, you need to place an order, and the laboratory will send you a sample kit, which will contain 2 cheek swabs, which you will use to collect cell samples from the inside of your dog's mouth. Now, it's important to remember that dogs like to grab things with their mouth, and if that thing is organic in nature, so it could be food, or maybe it was the ear of another dog when they were playing, there may be traces of contaminant DNA in your dog's mouth. That's why it's best practice to separate the dog you want to test before you take the sample, and make sure that you wait some time after the last feeding. To collect the sample, you will take the cheek swab and you'll gently rub it inside of your dog's mouth between the gum and the cheek. Then you're going to package the samples in the provided tubes, as described in the instruction leaflet that will be provided in the sample pack, and then send it back to the lab using the envelope provided. The lab will extract your dog's DNA from the cheek cells you have collected, and then they will interrogate very specific locations in your dog's genome to identify whether your dog has any copies of the mutations that you are testing for. They'll print out the results on a certificate, which is then sent back to you and to us as well. As I mentioned earlier, different labs and different authors may use different coding for the particular genotypes. So it is always important to check what the labeling is used by a lab which performed your testing. However, majority of the commercial testing labs nowadays are moving toward a simple, clear, carrier and affected coding of the results specifically for the recessive conditions. So a clear result means that a dog has no copies of the disease-causing allele, and it will only ever pass the normal allele to its offspring. So we would say that it's homozygous normal. A carrier dog has one copy of the allele, and while in recessive conditions this dog may be healthy himself, he has 50% chance of passing that disease-causing allele to any of his offspring. An affected dog has 2 copies of the disease-causing mutation, and this dog is either already sick or he will develop the condition in the future. It will also pass a disease-causing allele to all of his offspring. Currently, majority of the DNA tests examine the dog's genotype at the exact location of the disease-causing mutation. We refer to these tests as direct mutation tests. And this is to distinguish them from so-called linkage tests. Linkage tests are used when the exact disease-causing mutation is not known, but we know that it is very close to some neutral marker which co-segregates with that mutation. This was the first type of the tests that were developed in the 90s, when our knowledge of the dog's genome was relatively poor. But we still have to use such tests sometimes, as some bits of the dog's genome possess properties which make them very difficult to genotype. And in most cases, linked marker tests are relatively reliable, but we do need to remember that with linked tests, there is a risk that the linkage between the marker and the causative mutation may be broken over generations. So we might start to see dogs which have the marker but not the disease allele, or vice versa. So now that we know the theory behind DNA testing, let's see how we can use it as dog owners. I'm sure that just as myself, you spend so much time with your dogs that you think that you know them through and through. But sadly, many of the diseases which affect our dogs are late onset. So this means that our perfectly happy puppy and healthy puppy may develop crippling conditions few years down the line. A good example of this is primary open-angle glaucoma. And it is a condition in which the pressure of the fluid contained in the eyeball increases gradually over time, with typical age at onset of around 3 to 6 years. Initially, primary open-angle glaucoma doesn't cause pain to the dog, so it is very, very easy to miss the early stages of the disease. By the time we start to notice clinical signs, sadly, the pressure within the eye has caused an irreversible damage to the eye nerve. However, if we know that our dog will develop the condition in the future, we can administer early treatments which will considerably slow the progression of the disease. So if our DNA test tells us that our puppy is homozygous for the mutation, we can start the conversation with our vet to make sure that we apply the treatments at the optimal time to ensure that the condition is managed in the best way possible. Another example is a mutation causing multiple drug sensitivity. This particular mutation is less related to age, but more to the particular events in your dog's life. So the mutation which causes the multiple drug sensitivity alters the way that the dog's brain is protected from specific chemicals. So that means that drugs which in other dogs can save lives and are perfectly safe, in those dogs that are homozygous for this mutation, those drugs may cause a range of neurological symptoms, in extreme cases even leading to death. This mutation is particularly common in herding breeds such as border collies, and many vets are already aware of this, so they may suggest that you test your border collie before any procedure is done. But knowing whether your dog is homozygous for this mutation and letting your vet know the result so that they can adjust your dog's medication is such a simple way of avoiding a really horrible reaction in affected dogs. Another example of a condition like this is exercise-induced collapse, which affects some Labrador retrievers. Dogs which are homozygous for this mutation may suffer neurological issues, including collapse during and or following intense exercise. The severity of this condition varies, so some dogs will be able to tolerate mild exercise, maybe even moderate levels of exercise, while others won't be able to tolerate any exercise at all. There will be things like temperature, humidity that will affect this as well. Knowing whether your dog is homozygous for this condition, we can ensure that he gets the safe intensity exercise, but also coming from the perspective of a dog owner, if you know whether your dog is at risk of the exercise-induced collapse, you can prepare yourself mentally and physically to help your dog if they start to show symptoms. So by DNA testing, we are looking not at the dog as he is today, but at the dog that he may become in the future. So with this knowledge in hand, we can tailor their exercise, diet, and treatments for their unique genetic makeup. In humans, this approach is termed personalized care, and we are seeing that it is becoming the method of choice for the progressive doctors. But of course, the impact of DNA testing is even more important in the context of breeding for health. Let's go back to our case study, where our breeding prospect has a sister which produced affected puppies. And so we are worried that our dog may be a carrier. We've already discussed possible scenarios we could employ before DNA testing became available, and we know that none was very efficient. Now, thanks to DNA testing, the situation couldn't be any simpler. We collect a DNA sample of our dog, and then within a few weeks, we know whether he is a carrier or not. But let's take this a step further. What do we do with this knowledge? Well, this will partly depend on our goals. If our goal is to avoid producing affected puppies, then the situation is fairly simple. If our dog is clear, then we can breed him to any other dog, as our dog will only pass the normal allele, and so even if the other parent is affected, the resulting puppies will be carriers at worst. If our dog is a carrier or affected, although we know that from the pedigree this dog should not be affected, we can only breed him to clear mates, as any other combination would risk producing affected puppies. But perhaps our goal is to avoid producing affected puppies, but also slowly start breeding against the mutation. In such cases, we might decide that we might not want to use the affected dogs in breeding. And if we use carrier dogs, we might genotype all the puppies that they produce and keep for further breeding only clear dogs in the next generation. So we are gently moving towards breeding more clears and less carriers. Finally, our goal could be that we want to breed the mutation out as quickly as possible. In such case, we would breed our dog only if he was clear. We wouldn't breed a carrier or affected dogs, as they would both produce carrier puppies. So how do we choose our strategy for the use of DNA testing results in breeding? There are several questions we need to ask ourselves. First, we need to find out what is the frequency of the mutation in our breed. We need to remember that any form of selection results in a reduction of the genetic diversity. If there is only a very small number of dogs that are clear from the condition, then we must not use the intense selection strategy, as this will drastically reduce the genetic diversity of our breed. Further, we are going to spread the novel mutations unique to those few clear dogs. And the bad news is that because they are novel, there is no test we could use to safeguard against those. Secondly, we should take into account the DNA test results as part of a balanced breeding decision. So, for example, we might want to assess the severity of the condition that our dog potentially carries against other health test results of this dog. So if our dog is a carrier of a late onset, low impact condition, but is clear for severely debilitating diseases which are common in our breed, we might want to decide to ease on the selection against a milder condition to prioritize the severe condition first. So, as you all know, the Kennel Club collects the DNA test results in our database, which you can then access through our health test results finder, so you can find the best mate for your breeding dogs. But we don't just record these results, we also use them in research. So research and monitoring of particular mutations is crucially important for the efficient management of the breed priorities. So if we look at Labrador Retrievers, for example, we currently record 7 DNA tests for this breed, alongside recommended hip, elbow and eye testing. And on top of that, of course, we have other traits that we want to select our Labradors for. We want to make sure that they have the correct and stable temperaments. We want them to look a certain way. And we also want to keep an eye on the genetic diversity of the breed. So which of these breeding goals is the most important? When we look at the specific conditions, as we've said before, one of the important parameters is the mutation frequency. It tells us how many reproductive cells in a population carry the mutant allele. From this, we can estimate what is the expected percentage of carrier and affected dogs, and that gives us an indication how strict we can be with our selection. So, for example, if mutation frequency is 10%, then only 1% of the dogs will be affected with the condition, but nearly 1 in every 3 will be a carrier. So the higher the mutation frequency, the more gradually we need to select against it. Otherwise, we would be losing a huge proportion of the genetic diversity. But of course, looking at the overall mutation frequency doesn't really tell us the whole story. We also need to see how it has changed over time as breeders start selecting against it. Here we have 2 examples of the change in the mutation frequency over time. In both cases, the initial frequency among tested dogs were very high. Nearly half of all the reproductive cells among the tested dogs had the mutant alleles. However, the trajectory of the reduction in the mutation frequency was very, very different in the 2 breeds. The first breed managed to get the mutation frequency to nearly 0 in the last 4 years, whereas the second breed is taking the selection process much slower, and after comparable time, the mutation frequency is still relatively high at around 10%. And I should add that this is actually preferable in this breed, as it is a very small numerically breed, so gradual selection is the smart choice here. Of course, another very important question we need to ask is how well is the overall breed population represented by the tested dogs? So by combining the DNA test results with the pedigree data that we hold in our database, we are able to check this. On the left, we see the proportion of the puppies produced after tested parents for one of the tests we record. We see that in this particular case, this proportion is great, with nearly 100% of the puppies produced after sire with known genotype. The plot on the right shows the proportion of dogs born with known genotype in the same breed. And of course, ideally, we would like to know the genotypes of all dogs in the breed. However, this already gives us an indication that our estimates are relatively representative of the wider breed population. These are equivalent plots for another test in another breed. We see that the proportion of dogs proportion of puppies born after tested parents and proportion of dogs born with known genotypes are both relatively low. This tells us that we haven't tested enough dogs yet to have a realistic assessment of the wider breed. So the mutation frequency, its changes over time, and coverage of the population are all parameters we want to examine when establishing where a particular condition sits in a list of priorities for a given breed. This type of research drives the strategies for the overall breed improvement, which are then used in a range of initiatives such as breed health and conservation plans. Now, the value of DNA testing is unquestionable, and we are delighted to see that in majority of the breeds and across range of conditions, DNA testing has enabled breeders to significantly reduce the frequency of the causative mutations. A few years ago, my predecessor, Dr. Tom Lewis, and Dr. Catherine Mellers from the Kennel Club's Genetics Center published a paper on this exact subject, where they have examined in details changes in the mutation frequency for 8 conditions. In all cases, it was evident that selection with the use of DNA testing is very, very efficient. One of the very nice examples of that is the PRA RCD4 type in Gordon Settler, so progressive retinal atrophy. This mutation was first discovered by the team at the Kennel Club's Genetics Center when they were still located at the Animal Health Trust. And since then, the breeders have picked up the testing in vast numbers, with now more than 80% litters born after tested parents, and the genotype known for 60%, more than 60% of the population. The mutation frequency in the initial study population was estimated at 37%. Based on the records that we have had for the early dogs, it could have been even a little higher. But thanks to the use of the DNA testing, breeders have successfully reduced it to about 5% nowadays. And we see similar patterns across breeds and conditions. There are some variables. Obviously, the first one would be the length of time since the test publication. The second one would be the initial frequencies and the uptake of testing for specific conditions. But the general trends that we are observing are very encouraging, and in vast majority of cases, we see a drop in the mutation frequencies. So let's summarize what we have learned about DNA testing. DNA testing is a very basic tool for smart and ethical breeding. We can DNA test our young dogs with results available within weeks, long before we need to make a decision on whether we want to breed from them or not. It doesn't hurt the dog. It's easy to cheap, and it's easy to do, and it's relatively cheap. DNA testing provides us with an accurate statement of the dog's genotype. It takes guesswork out of breeding, even if we know very little about the relatives of our dog. Further, it tells us whether our dogs are at risk of developing a late onset condition, so that we can provide them with the best possible care. And of course, DNA testing your own dog has also huge benefits for a wider breed population, because those results contribute to the complete picture of the breed health, which then are used in further research and help us shape the most efficient strategies for breeding for health. Now, very quickly, I'm aware of the time. As promised, we are very quickly moving to the related subject of DNA profiling and parentage verification. As we've said at the beginning of this webinar, not all mutations will have an impact on the protein. If they don't have an impact on the protein, they are unlikely to be selected for or against. As such, they tend to accumulate over generations, with related individuals having higher degree of similarity in those neutral mutations than unrelated individuals. So we have known that for quite some time that a set of such markers can be used to uniquely identify an individual. The genotypes an individual has of those markers can be termed a DNA profile or a genetic fingerprint. And this technology was particularly developed for the forensic genetics, where the accurate identification of either criminals or victims is, of course, of utmost importance. It was then quickly developed into commercially available paternity tests for people. And in the last couple of decades, DNA profiling is used very widely to track livestock products. So, for example, to ensure that the meat that we buy in the supermarkets is actually coming from ethical and sustainable sources. DNA profiling is also widely used in horse breeding, and it has been available as a product for dogs for quite a few years. But the initial DNA profiles used in dogs were based on microsatellite markers, which are tandem repeats of a very short DNA sequence, and they are very highly variable. The old DNA profiles were constructed using just 22 microsatellite markers. Since 2020, the International Society for Animal Genetics has issued a new standard for DNA profiling in dogs, and this new recommended marker panel is based on around 230 SNP markers. Unfortunately, because the new panel is using a different technology and importantly examines different set of markers, the old and new profiles are not compatible. That means that if we would like to use profiles in parentage verification, we will have to use the same type of profile for both the parents and the tested puppy. Currently, it's still possible to obtain the old microsatellite markers in some labs. However, we should expect that over time it will become more and more difficult as labs adjust their products to the guidelines issued by ISAG. So the benefits of the DNA profile are that it is unique and a permanent form of identification. It cannot be lost because you have your dog in there. It cannot be manipulated and it can be destroyed. Further, DNA profiles can be used to verify the parentage of your dog. And there are multiple reasons why we might want to verify the parentage of our dogs. And of course, we are trying to avoid pedigree errors. Sadly, they are a real aspect of any breeding across all species, including our own. There are multiple causes for pedigree error. First and probably most common is an accidental mating. Some of those are known to breeders, others happen without breeders' knowledge. Some of those will result in multi-sire litters. So of course, we are always worried about fraud, where someone may try to raise the value of their puppies by stating that a different sire was used. And then also there are very simple errors in recording, or for example, if artificial insemination is used, I'm sure that you have heard those stories where the semen straws were mixed up. So let's very quickly see how parentage verification can work in the case of accidental mating, which happened unbeknownst to a breeder. Let's say we have produced a litter of puppies from black parents. We know that the dam carries chocolate, which in this breed, well, in all breeds, chocolate is recessive to black, but there were no recorded chocolates anywhere in the pedigree of the sire. As such, we assume that he doesn't carry chocolate, and we expect all puppies to be born black. And yet, in the litter of 5 puppies, we have produced a chocolate puppy. So the problem is that the neighbor has a chocolate intact male. So we start thinking, if we want to be sure whether the sire that we have chosen for our bitch is the sire of the puppies, the best solution for us is to obtain the DNA profile on the sire, the dam, and the chocolate puppy, and compare them to verify the parentage. So the DNA profiles using SNP markers state what are the genotypes that an individual has at the marker locations in the genome. So we see the old favorite ACGTs. We know that a puppy has to inherit 1 allele from the dam and 1 from the sire. So we go down the list and search for any particular mismatches. In this very simplified example, we see an issue in markers 3 and 7. Let's first look at marker 3. We know that the puppy has inherited 1 G allele from the dam, but where does the second G allele come from? The sire is homozygous for T allele. Apologies, my puppy is making a lot of noise right now. The sire is homozygous for the T allele, so it couldn't have passed a G allele to the puppy. When we look at marker 7, we see a similar situation. 1 of the T alleles must have come from the dam, but where did the puppy get the other T allele? The sire is homozygous for A allele, and so it couldn't have passed the T allele to the pup. As a result, we have to sadly pronounce that the chocolate puppy could not have been sired by the dog we hoped. The parentage verification tests are based on the exclusion. So that means that if we see discrepancy between the potential sire and the offspring, the laws of inheritance gives us 100% confidence that a given dog could not have sired that puppy. With this knowledge in hand, we now have a conundrum. Are any of the puppies in this litter sired by the intended dog? In the past, there was no way that we could answer this question, as we couldn't tell which puppy was sired by which sire. We wouldn't be able to register the puppies with the correct pedigree. But now, thanks to DNA profiling and parentage verification, we can. First, we need to obtain the DNA profiles for all the puppies in the litter. And if we just want to check whether they were sired by the black sire, that might be enough. If we would also like to check them against the neighbor's dog, we might want to profile him as well. And as a result of this, we will be able to separate our puppies in our litter into 2 half-sibling families. So with the parentage verified through the DNA, we can accurately and truthfully register our puppies with the correct sire record. So in conclusions for the DNA profiling, DNA profiling gives us a fast, cheap, and permanent method of verifying the identity of our dogs, and it gives us an undisputable proof of parentage where needed, and it provides an assurance on the traceability of dogs that we breed and own. And with this, I will very quickly give the virtual podium back to Charlotte so she can tell us all about the new DNA testing services that we have just launched at the Kennel Club. Charlotte, over to you. I'll skip to the next slide.
Thank you, Joanna, and I think you've earned a well-deserved drink there after a long presentation, but a very, very interesting one. Thank you very much for everybody for attending. We are going to take just a minute to give you some information about the DNA testing services, and then we're also going to invite you to ask any questions of Joanna. We have had some questions in advance, so we will go through those as well. So just to confirm for anybody who's not seen the press release or the information that's gone out recently, we have now launched the Kennel Club DNA testing services. We are offering single or breed packages for 80 breeds. All of the information is available on the shop. We are offering a discount on the health testing side. There is a £20 off discount for breed specific bundles for the whole of November. It's also really important to note, just on the back of Joanna's presentation, within our health breed bundle packages, we are offering the DNA profile included at no extra cost. So it's a really, really good opportunity if you do use our service to actually have the DNA profile included. In addition to that, for breeds that we're not able to offer health testing for at this time, we do offer independent DNA profiles for those who would like to have their dog individually profiled. We also have a parental verification service, which is now available. So there's a huge amount we've added from the 1st of November. We will work to look to add testing for further breeds as it becomes available, and we will work to enhance our menu wherever we can. There is a short film available on Facebook, on the Kennel Club's YouTube film, predominantly aimed at people that are not familiar with DNA testing. And just to really reiterate that if you can share this, really spread the word about the importance of DNA testing. We are one of many providers, but we are definitely one that people should consider if they are new or if they're interested in breeding or thinking about DNA testing their dogs. So that's our little piece on the DNA testing services, and I'm going to go straight to the questions. Just to start by saying, if we aren't able to answer your question this evening, that's not necessarily mean that we don't want to answer it. We're happy to speak to you offline if we're not able to get to your question. So first question is, why is the Kennel Club not supporting the Cambridge Genetic Services Testing and Research Centre in Cambridge? I've used them this year. They were very efficient and cheaper. And there may be some additional costs to post swabs to an EU country. So I think we'll just take this really briefly. The Kennel Club Charitable Trust completely supports the Kennel Club Genetic Centre, which is now based at the University of Cambridge. It was previously based at the Animal Health Trust. The Cambridge Genetic Service Testing Service is completely separate to that centre. Now, some of the same colleagues and colleagues from the Animal Health Trust still work within that service, but it is entirely separate and it's managed by the University of Cambridge. So just to kind of confirm any concerns, that we do work very closely with the Kennel Club Genetic Centre team, and we will continue to do so. To pick up on the swabs going to an EU country, We've actually worked to keep the cost down as much as possible. Our laboratory for the Kennel Club DNA testing services is Weatherby's Scientific, which is based in Ireland. If you want to have a look at their website, it's Weatherby's Scientific, and you'll be able to see the testing they offer for other species as well. But we've worked really hard to keep the costs down. The costs that are on the Kennel Club website are inclusive completely of postage, packaging, VAT and everything else. So hopefully that's answered that one. Joanna, thankfully a question for you. The DNA company that I use to test for over 215 disorders, some are not viable to my breed, but they are DNA single cell tests when available, when not a marker test is used. Do you offer an all condition testing package for breeders and their puppies? And I apologise if I've got that question horribly wrong.
No, thank you very much, Charlotte. So yeah, it's a very valid question, because I think that for all of us, when we see an offer 200 tests versus free tests, we will think that the offer that has more tests included will be more valuable. But we are going back to this question about the verification of the causality of the mutation on the disease in a particular breed. So we are not offering, and we are not planning on offering a test that will show you all the mutations that have been detected in dogs across all breeds in any particular breed, because we know that this can be quite dangerous. So it's fine if someone has a very educated approach. They can look at the results for 200 disorders and say, I know that these conditions are not relevant to my dogs, so I'm not going to pay attention to the fact that apparently my dog is a carrier of this particular condition. I'm going to concentrate on the breed relevant testing. But unfortunately, there will be people that won't have that in-depth knowledge of how to deal with the health test, DNA test results for these conditions. And they might, unfortunately, with very good intentions, start to select for a mutation that actually in your breed has no impact on health. So this would be the exact scenario where we are reducing the genetic diversity for no reason. We are not improving the health. We are just reducing the genetic diversity. We are removing from breeding dogs that actually have no problem and they could be safely bred from. So we have opted to offer a product which only contains the tests that have been verified. We know that they have been found in the breed. We know that they cause the disease in the breed, and we feel that this is a much safer approach to breeding for health with the safeguarding of the genetic diversity. I hope that's answered the question.
We have another question from Kim. Will we need to test genetically eye-clear litters after the second or third generation?
So I believe that this question relates to the hereditary status. So for those of you who don't know, the Kennel Club has been assigning hereditary status where we can predict we can state the genotype of the offspring based on the genotypes in the parents. So as we've said in the presentation, if we have both parents that have been DNA tested clear for a mutation, clear of mutation, they can only pass the normal alleles to their offspring. So the offspring has to be clear as well, by definition, by the laws of inheritance. So in those cases where we used to have both parents that were clear, we would assign a hereditary status. In most cases, this was hereditary clear. In theory, we could also assign the hereditary carrier if one parent was affected and the other parent was clear, or we could say that a dog was hereditary affected if both the parents were affected. But obviously, that was a much less common occurrence, luckily. So this process uses the laws of inheritance, and by the laws of genetics, it's definitely as it should be. However, we are also relying on the fact that the pedigree is errorless. We are assuming the parents are as listed on the pedigree of a particular puppy. And unfortunately, we know that sometimes pedigree errors happen. And if it happens at some point in the pedigree, and we have several generations of hereditary status, we could be proliferating the errors of the hereditary status where in fact dogs are carriers or even affected. So to deal with this risk of pedigree errors affecting how the hereditary status is actually relevant to particular dogs, the Kennel Club is going to introduce a limitation on the number of generations where the hereditary status will be applied. So it will be limited to 2 generations. So that means that if I DNA test my dog and I DNA test the mate of my dog, the puppies will get the hereditary status. The grandpuppies will get hereditary status, assuming the parents and grandparents on the other side are also clear or hereditary clear. But in the next generation, so it will be great grandpuppies, they would have to be DNA tested anew. The one exception to this rule is going to be if we have parentage verification on all the individuals in the pedigree going back to the DNA tested dogs. If we have the DNA parentage verification, sorry, I don't know what I said, then we are sure that the pedigree is exactly as stated. And in that case, we are confident that we can continue to assign the hereditary status for the upcoming generations as long as we continue with the verification.
Thank you. Lynn has asked, would it ever be possible for DNA testing to take the place of microchips?
So that's an interesting question. From the point of view of genetics and technology, whether it could replace microchipping, yes, absolutely. There is no reason why it couldn't. And in fact, DNA profiling has some benefits over microchipping because you cannot remove DNA profile from a dog. With microchip, you can. But there are some wider questions around this particular question. So I would say that it's probably best to discuss this with DEFRA, maybe. I wouldn't want to give the answer whether this is the direction that the government is going to go in. But from the point of view of technology, absolutely yes, it could happen.
Thanks, Joanna. Janice has asked, does DNA testing in puppies flag up genetic defects in that particular breed?
So yes, we just have to be clear on the specificity of the DNA testing. So DNA testing tests for specific conditions. If someone decides to test for 4 out of 5 conditions for which DNA testing is available, then we will have information about 4 out of 5 conditions. We are not going to know anything about that 5th condition that the breeder decided not to test for. And equally, if there are conditions in the breed that don't have a DNA test available, or a good example would be hip dysplasia, we don't have a DNA test for hip dysplasia. So unfortunately, for those conditions, DNA testing of the puppies is not going to provide us any information. So it is going to inform us whether the puppies have any of the DNA testable conditions in that breed. It's not going to tell us whether the puppy is going to ever develop any condition you could imagine.
Hey, thank you. Declan has asked, at the moment there are limited breeds that are encouraged to do the DNA testing. Is this list going to expand to all breeds in future, or will it remain limited?
Yeah, it's. I think that Charlotte already mentioned that we are working on expanding the list. Absolutely, as a general rule, I think that's. I'm safe to say that we do recommend DNA testing in any breed where relevant DNA testing is available. But we have to go through the evidence base before we add those tests to the list of the tests that we offer. So as you can imagine, in large numerically breeds, it will be slightly easier because it's just easier to find study subjects for scientific publications in large breeds. And some of the smaller numerically breeds, we haven't. The researchers haven't published any papers yet in those breeds. So it might take a little bit more time before we gather the evidence that is necessary to actually say with confidence that yes, this DNA test is going to be valid for this specific breed. But yeah, I would like to kind of go back to the point that I've made before, that we are very much relying on the collaboration with the breed communities. So if there are any tests that you think that should be included for your breed, my first suggestion would be to get in touch with your breed health coordinator to discuss this, because it might be possible that we have already discussed this test, but for whatever reason, let's say, yeah, the validity, the causality of the mutation has not been confirmed yet in your breed, and the breed health coordinator is working on gathering the data. Pardon me. Or in some cases, it could be that we have missed a test. We are only human, and there is 222 breeds with hundreds of different conditions. So if it turns out that you reach out to the BHC, the breed health coordinator, and you discuss that actually there was no previous discussion about this specific test, then I would encourage you to work with the BHC so that you submit a DNA test recognition form, and then we'll get the conversation going.
Joanna. So Deirdre has asked, should there be a requirement for anyone selling a pup to genetically test both parents before the sale of a puppy, or is trying to aim for genetic perfection a dangerous game?
Well, I think that this
on that one.
This is a really good question. It has two parts to it. I will try to go for the first one first. Yeah, I think that all of us that are in this webinar, we deeply care about dogs, and we deeply care about the health of our dogs. So yeah, I think that we all share the same dream that at some point in the future, health testing will become a standard for anyone breeding dogs. But there are some caveats to this. Unfortunately, it's not as simple as, yeah, let's do this. Let's all test our dogs. I should probably start from saying that at the Kennel Club, we don't have any legal powers over what people are doing with their dogs. We can lobby, we can encourage, we can educate, but legally we cannot tell people that they cannot breed unless they health test. Of course, we could make health testing a part of our registration process. And yeah, it sounds like a very simple solution, but there are a couple of caveats here. For this to work, we actually need to know that we have a support from the majority of the breeders and puppy buyers. The whole community of dog owners has to understand how important the health testing is. Because if we don't have that, if half of the population still doesn't believe in health testing, there are two possible things that could happen. The first one is what we have seen with other dog legislation, like breed-specific legislation. I've just gone blank. It is BSL, breed-specific legislation, right? We know that if a legislation is introduced and people are not on board with that legislation, some people will find a way to go around that. So we would have false health test results, we would have false pedigrees. If people don't believe in it, they will find a way to cheat the system. The second scenario is that there could be a group of people that will decide, okay, we don't care about KC registrations, we are going to do it on our own. And either they will open their own registering body, or they'll just do that without any supervision at all, let's say. And I personally see two problems with that. The first one is, I think that we are all dog people here and on the webinar. So if we are being honest with each other, if there will be any sort of divide between the Kennel Club and any new organization that would be formed, that will lead to a very deep divide. And that means that any sort of resources that we as the health team are going to prepare for people, just like the webinar that we are having today, this will never reach this other group of people. And more importantly, it's not going to make any impact for the dogs that they are breeding. So that's one issue. And the second issue from the point of view of genetic diversity is that we are going to cut our breed populations in half or maybe more. We already have some breeds that are in trouble from the context of genetic diversity. If we cut this number even smaller. If we cut the numbers of dogs that are actually actively breeding within the Casey registration system or in the other registration system, it's going to create really serious risks for many of those breeds. And the second point about the perfection, breeding for perfection, we should all strive for the best, but we should definitely keep in mind that we are dealing with biological organisms. So achieving perfection is most likely never going to happen. So maybe we'll leave that question on this, because we still have 5 minutes and plenty more questions. Sorry.
Thank you, Joanna. Thank you. So Stella has asked, I'm confident that DNA testing can confirm parentage of a puppy or that two dogs are siblings. Okay, so confirm parentage of a puppy or that two dogs are siblings. However, I'm cynical about the identification of crossbreeds of totally unknown parentage. How accurate is DNA testing for breeds of third or fourth generation crosses?
Thank you very much for this question, because yeah, it's definitely something that we haven't covered in the webinar, and it's something that we need to clarify. So the DNA profile and the parentage verification that we are going to offer in our DNA testing service are completely separate from the breed assignment tests that are offered by other labs. We do not do breed assignment tests. So all three of those products are using SNP markers. They are looking at the genotypes that dogs have at specific points in the genome, but then what we do with this information is completely different. So with DNA profiling, we have a simple statement. These are the genotypes that your dog has at these particular locations in the genome. With the parentage verification, we compare the DNA profiles, we look for the mismatches, and if we find a mismatch, that gives us a definitive answer that this dog could not have sired that puppy. With the breed assignment tests, what they are doing is they take I don't want to call it DNA profile because it's different, but they look at the number of marker locations in the genome, and then they compare the genotypes of a particular dog to a reference population for a particular breed. And then from this comparison, they derive a probability that this tested dog is actually coming from a breed. So we are looking at a likelihood-based result. Now, there are several aspects that will affect how accurate those tests are. But I'll be honest, because we do not offer that test ourselves, I don't really feel comfortable providing information about the tests offered by other labs. And when it comes to the accuracy, I would deeply recommend that people ask the labs that are actually offering those tests about the accuracy estimates that they have for their products. So just to kind of underline, we have the DNA profiling and parentage verification. We do not have the breed assignment tests, sorry.
Thanks, Joanna. We've got one more question that I'm aware of tonight. How important is it's from Francesco. How important is the COI, and how do you choose the perfect match according to COIs? And I'll say at this point, there may be a future webinar in this area, but if you want to take it briefly, Joanna, please do.
Oh, well, it's hard to take that one and give you a brief answer. It is departing a little bit from the subject of the webinar. So for those of you that do not know what COI stands for, it's the coefficient of inbreeding. And the coefficient of inbreeding estimates the probability that an individual will inherit 2 copies of a gene that are coming from the same common ancestor sometime back in the pedigree of that dog. So as a general rule, we advise that people choose matings where the COI is minimized. So we want to go as low as possible. Certainly, we want to keep the COI of a litter at the level of the breed average, or even better, below that average. However, again, there is no such thing as perfection in animal breeding. And when we think about the COI, we have to consider COI as part of the wider picture. So, for example, if you have a litter of 2 dogs that are having they would produce a litter that would have a COI of 0%. So from the point of view of the COI, it would be the perfect combination. You can't do any better than 0. But both of these dogs are carrying the same mutations. Then in that case, the testable mutations. So we DNA tested them, and they came back as carriers for the same, let's say, juvenile laryngeal paralysis. Looking at my own breed. So in that case, even though the COI is great, is perfect, is optimal, I would still not breed those dogs because I know that they could produce puppies that are affected with this specific condition. On the other hand, we could have dogs that have a COI that is not 0. Let's say it is about the breed average, but they have great health results for all the health testing that we recommend. So in that case, I would much rather use those dogs than dogs with COI that is a bit lower, but they are carrying specific mutations. So it has to be a part of the wider picture. Generally, we want the COI to be as low as possible. One quick caveat here. When we talk about the COI of 0, we have to remember that the calculation of the COI relies on the pedigree. So if we have an imported dog, the pedigree depth for this particular dog is going to be lower than for the national dogs. For the national dogs in many breeds, we have pedigrees that go all the way back to 1910, 1920. For the import dogs, we will have 3 generations of the pedigree. So this particular dog may appear as unrelated to the dogs that we have nationally, and so the COI of the puppies produced by that import would be around 0 or very, very low. But in fact, when you are breeding with imports, it is worth actually going back and testing the pedigree of that imported dog a bit deeper to make sure that actually there are no relatives that we haven't covered in the 3 generations pedigree that we have had, because the COI is based on the data that we have available. So I think that maybe that will do for this question for today.
Thank you, Joanna. Now, as far as I can see, and my technical skills will allow, I can't see any other questions, but I would like to thank you for a really detailed and really interesting webinar, and I hope that everybody's enjoyed it. I do have a few bits of information for anybody that's interested. We are going to be holding a webinar series next year, and we will provide information on those topics, hopefully by the end of this year or early next year. So please do look out for that. We'll have lots of experts like Joanna in different areas and with different things that affect different breeds. So we want to make sure that you have as much notice as possible to join those webinars if they're of interest. We also have a health and welfare conference on the 12th of November at Chesford Grange in Coventry. You can find information for tickets on the Kennel Club website if that's of interest. Joanna is giving a talk at the conference and will be there and available for your questions, which I'm now signing her up for. So please do think about attending the conference if that's of interest to you. All the information relating to Kennel Club DNA testing services is also available on the main page of the Kennel Club website. And if there is something that you wanted to discuss today, you may potentially didn't want your question to be read out, or you have a thought afterwards, or any feedback, we're always interested to hear what people have to say. We always want to try and enhance and evolve what we offer. So if you want to email health@thekennelclub.org.uk if there's any specific questions relating to specific breeds, then you're more than welcome to do so. And thank you very much for attending. It's 8:33 by my watch. So I'll say that's a good evening, and I hope everybody enjoys the rest of their evening. Thank you very much.